Hello, everybody. Thanks for coming. I am so pleased to introduce my friend and colleague, Dr. George Matsumoto. I got to know George when I was a graduate student studying at the Hopkins Marine Station, and he had just finished his PhD at UCLA. And we were converging on some of the new technology that was happening on the California coast, putting remotely operated vehicles in the deep sea. And I took a right-hand turn and went to the terrestrial ecosystems, but George stayed and became a senior education and research specialist at MBARI. He's one of their founding participants. And I'm just so pleased that he's here today. He's got a couple interesting things that he wants to share with you, which he'll talk to you about. But one is that this afternoon, he's going to be giving a seminar, special seminar for students at 4:30 over in Wrigley Hall. It'll be also on Zoom, but he's going to be introducing the MBARI summer internships, which are for graduate students and undergraduates. And we have some graduate students who have taken that internship will be in the audience. So if you want to send your students over, it's over in Wrigley Hall. And we will have pizza also. So without further ado, thank you, George, for coming to visit us here in the desert. Appreciate it.
Thank you for the invitation. Thank you all for coming. I'll try to do my best to beat the post-lunch fatigue and entertain you for the next 45 minutes or so. What I'd like to do is tell you a little bit about the Monterey Bay Aquarium Research Institute, which is where I've worked for quite a while now, and also tell you a little bit about some of the programs I run, and also Sharon mentioned I might want to share some of the research I am involved with. So I'm going to do all of that in 45 minutes. That's my goal. So to start with, this is a picture from the ocean looking at the Monterey Bay Aquarium Research Institute, and the humpback whales are not photoshopped in. We have deep water right in front of us, which means we get from time to time, especially in the summertime, feeding humpback whales right out of our windows, which luckily for me, I don't have an ocean view, so I have no problem concentrating. I can't speak about the people who did work. No, it's not quite yet.
Okay.
Oh, hold on.
Where's the pointer?
That's all right.
Sharon, is the Zoom open? It is.
No, nothing's working. Now the whole thing is frozen. It's because we practiced and it worked. There we go. Thank you. So I'm going to start off by acknowledging the fact that MBARI is located on land that does not belong to MBARI. And we're located on the unceded land of the Guanarine and Guachuron peoples. This area was home to a settlement called La Cuesta in the region of Calendarook, which means ocean home place. The native peoples of this area were taken to Mission San Juan Bautista and Mission San Carlos de Borromeo. And the native people who were taken to Mission San Juan Bautista are represented today by the Alma Mutsun tribal band. We were fortunate enough to be able to work directly with the Alma Mutsun Land Trust to develop this land acknowledgement statement. And just to put us in perspective, for those of you who aren't familiar with California or Monterey or Moss Landing, we are located in California, about midway along the California coast. And I'm going to use the laser pointer for those people online. And we are located here in Monterey Bay, which you can see down here in the bottom left, because of this enormous feature that cuts the bay in half right here. And this is an incredible feature that we call the Monterey Submarine Canyon. And the Monterey Submarine Canyon is the largest submarine canyon on the west coast of North America. It stretches down to 4,000 meters by the time you get 60 kilometers offshore. So 2.5 miles down. Within a couple of hours of leaving the coast. And what this does is it gives us incredible access to the deep sea on a daily basis. So Santa Cruz is up here at the top of the bay. Monterey and the Monterey Bay Aquarium are down here at the bottom, and Moss Landing and the Research Institute are right smack in the middle. And just in comparison, you might be familiar with the Grand Canyon, perhaps. Monterey Canyon and the Grand Canyon side by side, just to give you an idea about the size and scale, roughly comparable. But we know a lot more about the Grand Canyon than we do about the Monterey Submarine Canyon. For instance, we still have people who are arguing about how the Monterey Canyon was formed. not so much about the Grand Canyon. And I could spend probably a lot of time talking about MBARI, but I think the easiest thing to do is to play this short 5-minute video because it describes MBARI extremely well. And I don't have to talk, which I think is a good thing.
In 5, 4, 3, 2, 1. 1,200 meters.
Everything in the ocean has a story to tell. It's huge, it's dark, it's cold. How do you access this space? Simply by having a presence, you see things that no one has ever seen before. You discover animals that are completely new to science, or a phenomenon that no one knew was occurring.
You periodically come across something that is so magnificent that you didn't expect to see, and it's just breathtaking.
Tomorrow the winds will blow, they'll pump nutrients to the surface, and everything about the system will change. Where things are, who's there, how fast they're growing.
We're in a position to be able to share our discoveries and our love of the ocean in ways that were never possible before.
There are no departments of chemistry or biology or engineering for that matter. We are all mixed together, and that's very unique.
Each side can inspire the other to ask challenging questions and come up with innovative answers.
A scientist will know what they're looking for, but the engineer can illustrate the possibilities beyond what the scientist first imagined.
And that's the trick, is understanding those differences in language and how we put them together in order to realize the kind of device that we're trying to build. We marry that science and engineering partnership with our marine operations personnel. And the only way to make all that work is to have a group of dedicated professionals to operate the vessels, to operate the equipment, to recover it. It's really that three-legged stool, science, engineering, operations, that allow us to do the things that we do and work as a team. Oceanography is a contact sport. We have to not only immerse ourselves in that realm, but we also have to acquire material. We have to manipulate that material.
MBARI's here in Moss Landing because of the access to deep water that we can get to so quickly.
If we were to drain Monterey Bay, you would see a canyon equivalent size to the Grand Canyon.
So we get these deep, cold, nutrient-rich waters fertilizing the upper ocean. It gives us an incredible opportunity to understand that connection of the deep sea to our coastlines where most of our fisheries come from.
One of MBARI's guiding principles is to make our measurements in the natural habitat rather than hauling the animals out and studying them in our world.
It's important that discoveries we make have an impact and tackle problems like climate change, like ocean acidification, and exporting that knowledge, making it available to the whole world. One of the really unique things about MBARI is our partnership with the Monterey Bay Aquarium. We have the opportunity to bring fundamental science and technology and marry that with their amazing global reach that communicates important issues about the ocean to the public, to policymakers. We really want to be transformational as opposed to just incremental in the work we do.
Oceanography is in the middle of a robot revolution with platforms that can take us to places we couldn't be, at times we couldn't be there. It's really a fundamental shift in how we think about things.
We have new ways of assessing fisheries, better ways of managing precious resources. We're sitting on this mountain of highly curated information here that was being mined to extract quantitative information like who's there, how many, how do they relate to changes in the environment and so forth.
And that gives us an incredible ability to think about these things at a grand scale and to make those connections that are really challenging, but that's what we need to do.
Just by studying anything in the ocean, you are studying climate change.
The ocean is absorbing a tremendous amount of carbon dioxide and absorbing a lot of the heat that's being produced. And the question is, what does that mean for the ocean? What does it mean for the life that depends on the ocean? And What are the consequences in the future?
How changes at one step have implications through the entire ocean food web and atoms to ecosystems, and all the way up to humans on Earth.
I don't know anywhere in the world where you have this confluence of the place of Monterey Bay, the opportunity of science-engineering partnership, and the charge to do something that's high risk, long duration, unknown outcomes, and at the end of that, the expectation that you will just share it with everyone. You will essentially give it away.
Every time we answer a question, we have 20 new ones. Often we think we know what's happening, and then the biology surprises us.
It's really amazing how much MBARI has been able to be the ambassador for the open ocean and the deep sea.
That's why MBARI is here. We're supposed to do things that might not be possible by any other approach. I don't imagine there's another place quite like it.
That's a nice inspirational video about MBARI, just to tell you a little bit about us. And accessing the deep ocean has always been a challenge ever since ocean science has started. And one of the first ships that went out there to do a lot of quantitative work was the voyage of the HMS Challenger, which took 5 years to go around the world a couple of times. And if you ever have a chance, you could read the 20 volumes of work that came out of the Challenger expedition. It really is amazing how much work they did. But the type of work they did was using what we would call old-fashioned tools, except that we're still using the same tools today, right? Our technology for exploring the ocean really hasn't changed incredibly over the past 200 years. So they use nets, they use thermometers to measure the temperature, they used water samples to get water, water samples from discrete depths, and they learned a lot about the ocean. But in terms of the animal life, what they mostly learned about were the hard-bodied organisms, things like fish and crustaceans, shrimps, because those could be caught in nets. Things like squids or cephalopods or jellies, not very much in the Challenger expeditions because those don't collect well with nets. And so even after this long 5-year voyage, there was still a lot of the ocean that hadn't been explored yet. And that's where MBARI started. At the time when MBARI started in 1987, our founder and mentor, David Packard, who is also one of the founders of Hewlett-Packard Computing, he had a clear vision for how MBARI would tackle this challenge of learning more about the ocean. He was an engineer. He wanted engineers to participate in the challenge of exploring the ocean because he thought engineers had a great deal to offer. His vision was very different than most other organizations at that time. This is in the 1980s. Most organizations were starting to work on human-occupied submersibles, like the Alvin. And since then, there have been the Pisces. There have been a whole number of them, the Johnson Sealings. A lot of organizations were using their resources to put humans into submersibles to go down into the ocean to learn about the ocean using their own eyes. David Packard, his vision was to use a robot for a number of reasons. One is much more cost effective. Two, it's much easier to share what you learn because everything you see through the camera of the robot gets recorded and you could share that. Everything that people see from on board these human-occupied submersibles, it's up to them whether or not they want to share it or not. And that gets problematic if you really want to explore the ocean and share it with as many people as possible. And just from a personal viewpoint, I've been fortunate enough to go down in a human-occupied sub. I went down the Johnson Sealing. One thing that all the human-occupied submersibles lack are restrooms. So think about going down for a 12-hour dive, sitting in a seat that you can't get out of, and usually sitting right next to somebody else who's flying the vehicle, who also can't get out of their seat. And if you happen to be a woman, almost all the pilots were male. There's only one female Alvin pilot who got so tired of listening to the scientists talk about what they were finding, she decided to go get a PhD, and she is now a PhD researcher, hydrothermal venticologist, but very few women pilots. So MBARI started up in 1987. We are a nonprofit research organization. We have a staff of about 200 people, And what we're looking at, as that movie talked about, are some of the large challenges that we're facing in the ocean right now. Warming, oxygen depletion in the ocean acidification, coral bleaching. Things are changing in the ocean, both regard to the chemistry and the biology. What we have to do is document those changes. We have to start by finding out a baseline. So we spend a lot of effort doing seafloor imaging, trying to make sure we have a picture of what the ocean looks like when we started, so we could document any changes that might be happening. This is a hydrothermal vent flash. It's kind of cool because you've got the heated fluid puddling up underneath the ledge, and it looks like a sea underneath. And then it's overlapping and then going on up to the surface. This is all hydrothermal vent fluid. And this is in the Gulf of on Baja California. We use autonomous underwater vehicles. These are vehicles with nobody on board, nothing but computers to tell it where to go. We use these to map the ocean wherever we're going to send the remotely operated vehicle, the robot, because we need to find out where all the possible things we might run into are. And we want to map at much higher resolution than anybody else has. We can't map from the surface because that's usually 50 meter to 100 meter resolution. Our robot is smaller than that. We need to map at a much finer resolution. And so our maps are usually at the 50 centimeter or down as low as 10 millimeter resolution, all depending on how close we get to the ocean floor. And the ocean floor is still less mapped than the surface of the moon. We still know more about the surface of the moon than we know about the ocean floor. Because most of the existing bathymetry, most of the existing maps of the ocean floor are taken from shipboard sonar going down. And like I said, that's at 100 meter resolution. So for instance, the search for the passenger plane that went down in the Indian Ocean, 5 years they searched, never saw it. They mapped a lot of the ocean, but never found a plane. Just to give you an idea of how little we know about the ocean floor. By mapping the ocean floor and looking around at places, we find unusual things. This was just pure chance. We had an extra day at the end of a cruise. And Jim Barry here, who was studying corals, undersea coral gardens on Davidson Seamount off the California coast, said, you know, there's this weird little protrusion on the seafloor that we see in our maps. Let's go take a look. And that was called Sur Ridge. And Sur Ridge, it turned out, was an incredibly productive seamount, just covered in these lush corals and sponges, and is now one of our permanent study sites. We're using machine learning because one of the things we figured out very quickly is when you're recording video through your robots and you start the video when you put the robot in the water and you stop it when you take the robot out of the water, you're getting 8 to 12 hours of video every time you go out to sea. We go out to sea 160 days a year with each vehicle. That's a lot of video. We have 6 people in our video lab whose job it is to watch video and annotate it. What that means is that everything they see on the video gets logged and put into a computer database, along with all the information that the robot is collecting as well as the surface ship. It all gets tracked together. Everything we see, not just the things we're interested in, but things nobody's working on. It's all in the database. That takes an incredible amount of time. It takes 2 hours to annotate 1 hour of video. So if we're generating 20 hours of video in a day, that's going to take 40 hours of somebody's time to annotate. And we're recording video 150 days a year. So machine learning was a natural. And with machine learning, what we're hoping to do is be able to use computers. We want to train computers to do some of this annotation for us. And it's an incredibly complex system. It works much better on the ocean floor than it does in the midwater, just because in the midwater, there's so many things going by and so many particles, it's harder to train the computer, but these are some of the challenges we're working on. And so as you're flying through the midwater, you can see that the computer here is starting to identify things that you and I might say, well, I don't even see anything. What was that? All right. And in the video lab, the annotators are going frame by frame. They see all these things. And we've been working on training the computer to do exactly the same. Right now, humans are still better by a factor of, they pick up 10% more individuals than the computers do. That's still pretty good. Machines are picking up 90% of what a human can pick up. We want to do better than that. We're never going to put the humans out of a job. That's not our goal. Our goal is to make the humans' job easier by having the machines do some of the work for us. What are the robots that we're using? We're using the remotely operated vehicle Dock Ricketts. This is a 4,000 meter capable remotely operated vehicle. Goes down to 4,000 meters, can stay in the water as long as our pilots feel like staying in the water, usually a 12-hour dive. We have the remotely operated vehicle Ventana. Ventana is an 1,850 meter remotely operated vehicle. It usually goes out and does a 6-hour dive because it's on our, what we call our day shift. We go out a couple hours, we put the ROV in the water, we do a 6-hour dive, we pick it up and we come home. Everybody gets to go home for dinner. That's nice. So 6 hours with this vehicle, 12 hours with this vehicle. And then we built this little one we call the mini ROV. It doesn't even have a name yet. We haven't given it a name. It's just called the mini because it's tiny. Initially, we built it out of spare parts that we had lying around. Since then, we've improved it and we've beefed up the thrusters and the camera system and the electronics on board. And we use this as a remotely operated vehicle of opportunity. Both of these remotely operated vehicles are tied to our surface ships. They're big, right? I mean, Dock Ricketts is 8 tons, 16,000 pounds. It's not moving, right? It stays on the ship and goes down. Ventana stays on the ship. But the mini ROV, we could put in a shipping container, ship it anywhere in the world, put it on any ship that happens to be going out and has some deck space for us. And so this has become an incredibly useful tool to our researchers who want to work in other places in the world besides California. But we're also using other things. We're using gliders, which I know are being used here. We're using autonomous underwater vehicles like I talked about before. We're using wave gliders up at the surface. These are what we call the long-range AUVs, long-range autonomous underwater vehicles. Our big autonomous underwater vehicle will stay out to sea for 8 to 12 hours. That's its battery life. It's great for a day expedition. But if you want to spend more time out at sea, which is what we want to do, the long-range AUVs can spend 2 weeks, 3 weeks out at sea collecting data. Much smaller, can be actually launched by one person off of a whaler. Don't need a big ship to launch it. And then we're also starting to collaborate with Saildrone, because Saildrone is doing some amazing things all around the world. And we could utilize that platform to put some of our instruments on.
George, if I could ask quickly, the ROVs, how deep can they go?
The remotely operated vehicles, our deepest one is 4,000 meters, so 2.5 miles. Our middle-sized one is 1,850 meters. And the mini ROV is 300 meters. So pretty shallow, but we're working on making it deeper. So our main workhorse is the research vessel, the Rachel Carson. Rachel Carson, as I said before, is what we call our day ship. We can sleep people. We could sleep 12 on board, but we usually use it for day operations only. We leave the dock at 7 and we're back at the dock by 4. Nice day. We go out and do CTD, conductivity, temperature, density measurements, or we'll go out and do remotely operated vehicle missions. We have a tiny boat called the Paragon, which we use for mooring maintenance and launching some of our autonomous underwater vehicles that you could see there, as well as doing scuba diving and other things. And this is a nice boat because it's only takes one person to run. Very economic. And our new ship, we're just retired the Western Flyer this year. We're building a brand new ship, the research vessel David Packard. This is a picture taken at the launch in Vigo, Spain. They're finishing out the sea trials right now and building up the infrastructure up here. And that will be making its way through the Panama Canal and arriving at MBARI in August of this year. So we'll have a brand new ship and we'll put our big remotely operated vehicle on board, the Dock Ricketts. And we're going to launch it through this logo that you see here on the side. The logo is actually a door to a garage. So it just opens up and we launch the ROV over the side. Our vision is relatively simple. We envision a future where we better understand and can predict ocean processes. Seems easy, except the ocean's a big place. All right, it's involved in everything, and we want to understand that. And we want to do that by providing data and share that data with as many people as possible. So collaborating with Arizona State University and the School of Ocean Futures would be fabulous, because that way we could share what we learned. We want to make sure that whatever we do, we try to be as diverse, inclusive, and equitable as possible in everything that we work on. Our strategic roadmap is something we are updating right now. This is the latest draft, so you're getting it hot off the white paper, so to speak. Innovate and build. We want to make assessments, explore and protect, and inspire and engage. These are our primary goals. I'm sorry, assessments is part of that first one. Innovate and build, explore and protect, and inspire and engage. Those are our three main objectives. And MBARI is uniquely situated to do a lot of the innovation and build that other organizations can't do. We have an incredibly talented staff to do some of this work. Explore and protect. Explore is pretty easy because the deep sea is relatively unexplored. Right, so almost anything we do in the deep sea is going to be new. A little bit about some of the things I run. I've been at MBARI, as Sharon said, since 1996, most recently. So a few years. I run all the joint projects we do with the Monterey Bay Aquarium. So we work together and I'm the liaison between the two institutions. I run a seminar program, an internship program, teacher professional development program, adopt a float program, and I also do a little research on the side. Originally, when I took the position, it was supposed to be 50% research and 50% outreach. You could probably tell it's not quite there. It's probably more like 90% that and 10% that. Sort of like going into admin. But we're really excited in terms of the partnership with the Monterey Bay Aquarium. The Monterey Bay Aquarium opened up a brand new exhibition just last year in April of 2022. It's called Into the Deep, Exploring Our Undiscovered Ocean. And it's an amazing exhibition. And I hope all of you will get a chance to see it. You have 6 more years to see it. So no excuses, right? It's going to be open for 7 years. That's the plan. So you have 6 years left to come and take a look at it. But it's nice because they're featuring a lot of MBARI staff, unfortunately, including me, but we're working on getting all these pictures swapped out so that we could show some younger staff. But we have midwater animals on display, animals that we've collected. I'm going to talk about some of our collections in a minute. Also deep water collections, these deep sea corals, carnivorous tunicates, brachiopods, all kinds of things are all collected with our remotely operated vehicles and then conveyed to the Monterey Bay Aquarium where they're trying to keep it alive.
You exhibit live deep water coral.
Yes. Right? They're doing great.
I heard that.
See, you have to come visit. Right? So one of the fun parts are
When the rain stops.
I know when the rain stops, it stopped. Yeah. One of the fun things is that I've been working with the aquarium now because we can't collect all the animals using robots. And so some of the animals we actually collect using scuba. And so I've been fortunate enough to be able to work with the aquarium on some of their midwater collections for the new exhibit. Let's see if I can.
You might think that the
And this
world's
is a short
largest
video.
animal migration takes place on land, but in reality, it takes place in the ocean every single night. Millions of animals are moving tremendous distances from the deep sea up to the surface at night to feed. It's this constant process of moving up and down through the water as the sun is going up and down.
Right when the sun goes down, deep sea animals come up to the surface, which is amazing. They're coming to where we can see them with our own eyes. We don't need any advanced robotics to go down and search for them. We can actually go scuba diving and find them.
Tonight, our max depth is going to be 60 feet. A lot of the animals are located between 20 and 30 feet. Does anybody have any questions about the dive tonight?
Oh, good.
All right, let's have a safe, fun, and productive dive. Blackwater diving is a really cool technique that allows biologists or divers to get into an environment that's normally very foreign and hard to get to.
We're diving at nighttime because of this incredible phenomenon called vertical migration, where animals are moving up to the surface at night to feed. And then they go back down when the sun starts coming up because they're afraid of getting eaten. Imagine if you were hungry and you wanted to eat a meal and you had to run half a marathon up to the surface in order to eat your meal. And then when you were done eating, you had to run another half marathon back down into the deep sea to stay safe.
It's the right time to get in the water when the sun goes down, because that's when these deep sea creatures are coming up to the surface.
You're ready.
All these creatures that I study in the lab and have tried to learn so much about will be right in front of my face, finally.
For 25 years, I've been working at MBARI studying deep sea animals, and this blackwater diving expedition gives me a chance to actually see these animals in 3 dimensions right in front of me, and it's amazing. Okay. Blackwater diving, to someone who's not a blackwater diver, is hard to explain. It's hanging out off of a line underneath a boat where the bottom of the ocean may be 3,000 feet below you or deeper. It's all black above you, below you, all around. It's just black. You just don't know what's in the water next to you until you move your flashlight to highlight whatever it is that's near you.
Using this technique, we've been able to collect
The
animals
Monterey Bay Aquarium is opening up a deep sea exhibition,
with a deep sea.
and it's going to bring unusual deep sea animals that people have never seen before.
I'm going to keep going because that goes on for a little while. But that was in Kona, Hawaii. And so I'll be going on February 18th for the next collecting trip. So, you know, life is difficult. I don't get to do as much research as I want, but I get to go scuba diving in Kona, Hawaii for work. That's not bad. We have a seminar program. Our seminars right now are hybrid in terms of like most seminars these days, where people can come in and listen, and then we have a large audience listening virtually. I mention this because one of the things I'd love to do is get some of the faculty here in Arizona to come out and give a talk at MBARI. So you could learn a little bit more about what MBARI does, and we could learn about you and what you do, and see if we could develop some kind of relationship, some kind of partnership or collaboration. And if nothing else, you get to come out and see the deep sea exhibit, which is not bad. We do have a summer internship program, as was mentioned. Our summer internship program runs June, July, August. It's a 10-week program. It is open to undergraduates and graduate students. We tend not to take very many freshmen. We don't take very many fifth-year PhD students either, but somewhere in the middle. So even students who have graduated and are just thinking about graduate school, that's our primary target, right? Because you don't have very many other options. You can't apply to any REU programs because they only take undergraduates. But now you graduated. So now you're sort of aged out of the REU program, but you don't know what to do. We want you. Because for me, we run the program for the benefit of the intern. And our goal is to not necessarily get more people into STEM, although we think that would be nice. That's not our purpose. Our purpose is to help our interns figure out what they want to do with their future by exposing them to as many potential different careers and opportunities as possible during this 10-week program. This is a stipended position. Last year, it was $17 an hour. This year, it'll be $19 an hour. And next year, I hope to make it even more because the minimum wage in California right now is $15.50 an hour. We're trying to get a little further, much beyond that. I also run a teacher professional development workshop. So if you know anybody who's interested, if you know any teachers who want to come and participate in this, let them know about this. Our goal with the teacher workshop, this is K through 12, any subject. I don't care what you teach. Doesn't matter to me. You could come, and our goal is to get real data into the classroom. So we share with you stories from researchers who share their research, their data, and tell you how to work with their data. And what we want to do is get that data back into the classrooms so that students are learning real data and real world problems. And so this workshop is during the summer. The 2023 workshop will unfortunately have to be in Honolulu. So if you know any teachers who are interested in coming out, we'll take 20 teachers and it's all expenses paid. So the teachers don't have to pay for this. We cover all their expenses to come out. We're going to work on because they're going to make lesson plans. It's a fun workshop. All the participants who have done this have agreed. And then I also run the Adopt a Float program. These are biogeochemical floats, the Global Ocean Biogeochemical Array. MBARI has gotten external funding to build and deploy 500 of these global biogeochemical floats over the next 5 years. Started a year ago, so 4 more years. We're a little behind because of COVID and supply issues, but we're getting there. We're catching up right now. And so we are deploying all these floats, and what I've done with the float program is the floats get deployed, but they also get a logo on them, and they could be adopted by a particular school, an after-school club, or a homeschool group. Doesn't matter to me. Anybody can adopt a float. The school can adopt a float, right? And you can have your own float. And all that data, the floats last for 5 years. And they're a little bit different than the Argo floats. This is the Argo program. Argo floats, there's a lot of them in the water. Right, as of yesterday, 3,930 floats. And each black dot is an Argo float. Argo floats measure temperature and salinity, which is great if you're interested in temperature and salinity. But if you're interested in other things like pH or nitrate or chlorophyll, that's what our floats measure. All right, they do all the biogeochemical work. So it's a set of we add to the Argo float a set of chemical and biological sensors that get added to the float, and these floats then go out into the ocean. Oops, hit the wrong button. Oops, I thought I had a video showing how it played, but maybe not. But these will go out. They'll spend most of their life at 1,000 meters, 9 days. On the 10th day, they go down to 2,000 meters. And then they profile the water column from 2,000 meters all the way up to the surface, profiling all these measurements. When they get to the surface, they send that data to a satellite, and then they go back down to 1,000 meters and park. The float's lifespan is about 5 years. So it'll keep doing this every 10 days for as long as the float lives. So for 5 years, your float will be on the website, and you could search it by the float name that you've given it. So it could be the School of Ocean Futures float, and it'll be sending back data, which will be kind of fun. And we're also linked with the SOCOM program, the Southern Ocean Climate Change and Modeling Program, which is launching the same floats underneath down in the Southern Ocean. And that's kind of a neat idea because the Southern Ocean is really hard to measure in the winter. Storms and everything. And so, but these floats are measuring all the time, even when they're under ice, they measure. But what they do is as they come up near the surface, because they're also measuring temperature, when the temperature gets to 0, they're programmed to go back down because we don't want them to hit the ice because all the sensors are at the top. And so they'll go back down, and they'll just keep doing this every 10 days until they no longer detect sea ice above them. So we just had a float pop out last week that spent the last 3.5 years under ice. And it came up and downloaded 3.5 years worth of data. It was amazing. Great, great data set. And then my particular research focus is mostly on what we call gelata or jellies. And I'll study pretty much anything in the ocean. It's all fair game. There's a nice YouTube video called "There Is No Such Thing as a Jellyfish," but I'm not going to play it. I'm going to, you can see it for yourself on our YouTube channel. I'm going to talk about some of the things that I have been able to work on and describe. This is the first animal I ever worked on as a graduate student. This is before I started graduate school. My advisor told me, we need to make sure we're going to be able to work well together. So you're going to have to come out and spend 2 months with me in the Catalina Channel, diving just to see if we get along. And so we spent 2 months diving every day, which was a blast. The first animal I saw when I got on the water was this animal. And I asked my advisor what it was, and he said, I don't know. And so this is what we studied for 2 months. And we got 5 papers out of that 2-month period. And so I thought, God, graduate school's easy. I'm going to be done and out of here in just a couple of years. I was wrong. It took a little longer. But this is one of the first animals I was able to describe. It was an interesting one because when I gave it its name, one of the people reviewing the paper who revealed themselves, so not an anonymous reviewer, but she said, this is the name this is what you have to call it. Because this animal was actually described in 1945 by a master's student at Hopkins Marine Station in an unpublished master's thesis that's sitting in the library. And it's like, are you kidding me? How was I supposed to know that? But I went and got a copy of it and used the name that she described. Lucinda Stanford was a master's student at Stanford. Turned out not to be any relation to Stanford, but she called it Leucothea pulchra, and so that's the name we called it. And so we gave her credit for that. Since then, we've described a lot of other jellies in the deep sea, ranging a lot of ctenophores, because that's what I did my PhD on. These are comb jellies, no stinging cells, but also other types of jellies. Like this is a big skyphil medusa. This one we call Tiburonia gran rojo. Tiburonia, after the remotely operated vehicle Tiburon. This is a new subfamily, family, genus, species that we described. Gran rojo was actually because of my wife, because we were going to call it a different species name. But because I don't speak Spanish and she does, I was emailing her from the ship because that's where we were working on it. And I asked her how to say this word in Spanish, and she told me, and she asked me why, and I told her, and she said, you can't use it because it's rude. And she was right, because when we first saw this underwater, we gave this animal the nickname Big Ugly. And so I asked her how to say that in Spanish. She said, gran feo, but you can't use it. She said, what else is it? I said, red. So she said, call it gran rojo. So that's its official name. And my one regret, of course, is that I didn't acknowledge her in the paper, which I should have. Bad move. Any of you thinking about describing your species? Make sure you acknowledge your spouse when they contribute to your publication. So instead, what I did is I went in and made a wiki stub that went through the whole process. So if you want to read about it, it's all there on the Wikipedia page. But we found a number of jellies down there that are just unusual and strange and bizarre, but not just in the deep sea. Amanda Kahn was actually in the movie that you saw. She was the postdoc, but this is when she was an intern at MBARI. And she and I went diving at one of the local beaches when she was an intern, and we found this cool little jelly. I mean, that's a 5mm scale bar. This is not a big jelly. And we didn't know what it was. And it turned out to be not a brand new, because it turned out, of course, like most things, other people had seen this before, but had not formally described it in the literature. So this is a quote unquote new species of Staromedusa from the Northwest Pacific. And this animal we found in 5 feet of water, right? Not deep sea. There are new things in the ocean we're still learning, like this one. This is a box jelly. I love box jellies. Box jellies are amazing animals, but this box jelly is from California. California is not well known for having box jellies, but there is a population of box jellies in Santa Barbara. They show up regularly for 2 weeks every year, and then they go away. There have been probably a dozen papers of researchers who have done research studies using these box jellies, and they've been called 3 different species in the literature. And so we did a very thorough analysis and decided that this was an undescribed species, and we called it Cordia confusa, because people were confused about what species it actually was. And that confusion goes to almost everything we find in the deep sea. This is a paper we did recently on little red jellies. Turns out little red jellies are everywhere in the ocean, particularly in the deep sea. And all you see are little red jellies floating by, and that's what people say on the audio tapes. Oh, there goes a little red jelly. But nobody had ever really stopped to take a look at them. So we stopped to take a look at them and really differentiated them into all the different groups, found 3 different genera, one of which had been misnamed. So we corrected the name, and then we published it and then said, oh, by the way, there's still other ones that are out there that sometimes are red, sometimes are not, but we have no idea who they are. There's weird things out there. And it's now taken a lot more work to figure out what a new species is, because of course now you need DNA too, sequences. Here's mesophotic reefs down in South Africa. This was a fun paper, and my only regret was I didn't get to go diving in South Africa. Mark just sent me pictures and said, have you seen this? Do you know what it is? And we responded to him. And the last paper we published just last year is, oops. That video is not is a brand new species of a deep sea red jelly again, Atolla. We're calling this Atolla Reynolds eye. And it's named after Jeff Reynolds, who was the first volunteer at the Monterey Bay Aquarium. And he was the volunteer who, if you ever get to the Monterey Bay Aquarium, there's a whale skeleton hanging. He was the volunteer who slept overnight on the beach to protect that whale from people coming to take pieces of it, because the aquarium knew it wanted to harvest all the bones for that display. So he stayed overnight on the beach until the aquarium could get everybody there the next day to collect it. Pretty impressive. Oh, there's the movie. Beautiful, beautiful animal, 6 to 10 centimeters. So larger than the largest existing Atolla. The largest existing Atolla is called Atolla gigantea. So of course the thought was, well, we could call this Atolla gargantua, right? Turns out we have now found 2 new species that are larger than this. And so we just said, we're going to stop referring to size in the species name, because that's just never going to end well. But it's a beautiful animal, completely different than all the other Atollas, comes out molecularly, really clearly in a different group. So it's a really unusual deep sea jelly. And with that, I've taken my 45 minutes. I have time for questions. I have deep sea stickers for those of you who are brave enough to come in here in person. And I'll tell you that the deep sea stickers are also, there's a QR code on it. And if you go to that QR code, it'll tell you all about the animal you see here. And this animal on this sticker is actually an undescribed species. We have so many undescribed species. The aquarium deep sea exhibition actually has a tank that is labeled undescribed species. And we put animals that we know are undescribed species into that tank, because we know we're not going to have time to describe everything. MBARI has described a little over 250 species in the 35 years we've been around. We probably have another 300 animals that we think or know are new species, but we don't have time to write them up. It's crazy. Every time we go into the ocean, we find something new, some new behavior, some new animal, something is happening that we haven't seen before. So it's a blast. I'm hoping that some of you will come out and visit or come out and be an intern, and I'd welcome any questions. Yes.
Hi, this is really excellent talk, and I'm TV. I'm a faculty at School of Ocean Future. I'm using satellite remote sensing and machine learning to study the coral reefs and the water column and water quality. So I have 2 questions. The first question: how deep of the deep ocean coral, and do you see any bleaching or disease here, or do they doing good, or do you think it's comparable to some shallow corals? Any things like that?
Great question. So the first question is how deep are the deep ocean corals? And we find the deep ocean corals all the way down to 4,000 meters. Yeah, as shallow as probably the shallowest ones are probably 300 meters. But the really big colonies are sort of right in the middle, somewhere between 1,500 and 2,000. Absolutely no signs of bleaching on any of them, which is great. We like to see that. They do seem to be very sensitive to pH changes, which is some of the work we've been doing in the lab, just seeing how they're affected by pH. And they do seem to be very sensitive about, so that's something we're looking into further. Yeah. Yes, sir.
I'm Brent Scotting from engineering. In your biogeochemical flows that stay up for 5 years, how do you deal with instrument?
How did we deal with the what?
Is there a problem with the instrument drifting? The calibration?
The instrumentation?
Yeah.
Or the fouling, or we have very little fouling on the floats because they stay at 1,000 meters most of the time. We're using a bladder, a buoyancy bladder, an oil-filled bladder for the buoyancy to go up and down. It's a buoyancy engine, so it takes very little energy. The floats are not inexpensive. They're roughly $80,000 each, which is where most of the money is going for those. We're building 500 of them. And they are, unfortunately, they're what we call disposable floats, because at the end of life, as the battery starts to draw down, because we're talking to them every 10 days, as the battery starts to draw down, when it gets near the end, the final instruction is to go down. And the hull will then implode and everything will sink. We've done all the environmental impact reports for the National Science Foundation, and the impact of 500 floats going to the bottom of the ocean is much less than it would be if we were to try to get a ship out to pick them up.
So you calibrate them when they go out.
We calibrate them when they go out. So we calibrate them in the lab when we ship them. Then we send somebody down to the dock just to make sure nothing got bothered in the shipping. Then we put them on the ship, we put them in the ocean, and we get back the profiles every 10 days. And if something looks wrong with the data, we can quality control the data. We can change the calibration files so that we can adjust it if things start to look really wonky. And we are getting that right now with some of the floats we just launched. Our pH data that's coming back looks really bizarre. And so we think that we got sent the wrong calibration data. And so we're going to have to reprocess those profiles using new calibration data before we post them up on the web. But all the data that comes in from the floats goes to MBARI and then onto the web almost immediately. Yes.
Do you have nitrate sensors on this?
Nitrate, yeah.
Yeah, do you have issues with those at all? With keeping them stable?
We have been very fortunate. We have Ken Johnson and Yui Takeshita, who are sensor development people. So they're the ones who developed the pH sensor and the nitrate sensor. They've got the ISIS sensor. They're amazing, not only amazing scientists, but amazing engineers. And so what MBARI does with these programs is if MBARI is involved in the innovation part, the building, we don't want to build them for the community. So what we'll do then is we'll license it to the industry, who will then build it for the community. So all the sensors that we build, after we've built them, we license it. And then we and the community then buy it from the company that has licensed it. So for instance, right now we're working on a brand new oxygen sensor because we don't trust, I mean, we've been putting 2 oxygen sensors on because it seems so variable. So we've been building a new oxygen sensor that we hope to have on the floats and into production by next year. But it's nice, right? Because pH is normally something you have to calibrate frequently, right? Nitrate, the same thing. Oxygen, when you put a sensor on a float that you're not going to see again for 5 years, you can't waste time trying to standardize them all the time. It's just impossible. So you have to build a robust sensor that will last at least that lifespan. Yeah. Biofouling, again, where it's reduced because we spend most of the time at 1,000 meters. All right, and we don't spend very much time at the surface, just long enough to talk, and then they go back down. Yes.
What is the process to identify a possible new species? Why is it such a process that it's such a huge backflow? Is it something that takes a lot of time or a lot of effort?
To buy a what?
To define a species that might be.
Why is it so hard to describe a new species? That's a great question. So that first one I described, it wasn't very hard at all, right? I said we got paper out really quickly. It was relatively simple. I drew it. I compared it to all the other, especially for ctenophores, because ctenophores don't have any holotypes or paratypes because they don't preserve. So the only thing you could do is look at the literature and look at the pictures in the literature. So what I did is I drew it. I compared it to all the existing pictures in the literature, of which there were not that many. And then I said, well, I think it's different, so I'm going to call it a new species. You can't do that anymore. Can't get away with that. So nowadays, to describe a new species, it's not enough to do that. You now also really need to sequence it and show that molecularly, it's different from as many of the described species as possible. Like with Atolla there are 3 of the type specimens that don't exist anymore. Or they never did. And nobody knows who they are or where they are. So we're calling this a new species tentatively. It's pretty sure it's not any one of the other described ones because it's so big and so different from the descriptions. But we can't definitively say because we don't have the DNA from those early ones. but pretty sure it's a different species. So describing a new species now takes patience, persistence, and a lot of time, right? You have to read all the literature. You have to try to get access to any specimens, right? By loans or actually physically go to places like the Smithsonian or somewhere else. And you have to have either the expertise or a collaborator willing to do the DNA sequencing and analysis. For a couple of the papers, I did the DNA sequencing analysis myself. And then I took 2 years without sequencing, and all of a sudden I realized I can't sequence anymore because now they're doing a whole different type of sequencing. So one of my co-authors is a person who did all the sequencing and did the analysis, and she wrote up that section and put it into the paper. And that I'm fine with that. Boy, that's a lot of work. Yeah, so don't be afraid to collaborate on new species. That's, yeah.
Yeah, so at the beginning, you showed that one of the topics that MBARI works on is coral bleaching. Can you talk a bit more about that part? I mean, I love the jelly stuff. No.
Yeah, but you like corals.
But I like jellies as well.
You like corals. So,
Plankton, so you know, they're okay.
so Jim Barry started off as a shallow water, intertidal, subtidal ecologist. And because we had access to the deep sea, he shifted to the deep sea. It doesn't mean he's still not interested in shallow water. He is. Yui started off again on coral reefs. He's still very interested in coral reefs, and he still works on coral reefs. For both of them, we're interested in the effects of temperature change, pH change, and oxygen change, but not on their own. We're interested in all three variables changing at different times at different levels. So what we want to do is subject the organism, the coral, to different levels of each variable at the same time, because that's what's going to happen in the ocean. You're not going to change temperature and leave pH and oxygen the same. Everything else is going to be changing too. And most laboratory studies only do one variable at a time. And we want to do all three at the same time. And that's where our engineers came in. They built this incredible system so that it's pretty much hands off. We could put the coral into the chamber, hook the sensors up to it, push the button, and just let the computer control everything. Control the temperature and the pH and the oxygen levels. Right now we're working in chambers that are this big, but we also have been working on chambers that are 2 meters by half a meter by half a meter. So, sky's the limit. Yes.
Do you guys have any ongoing eDNA work in the canyon?
Great question. We're doing a lot of eDNA work, not just in the canyon, but we're also working a lot in freshwater lakes. So we've been collaborating with the USGS and Fish and Wildlife, and we've been helping them do surveys for exotic and invasive species. The eDNA work originally came from Chris Scholen's initial work on harmful algal blooms, where he was looking for the harmful form of Pseudonychia, which produces domoic acid, because morphologically, the form that has domoic acid and the form that doesn't have domoic acid look the same. So you can't do it on a microscope. You have to break the cell and pull it out and look for domoic acid. So he built a machine that would do that. And now we've retasked it to look for other things. And we've done it in what we call the lab in a can, which is what we've done in freshwater, where it's actually like a 55-gallon barrel that we could take out, deploy, and all the water samples run through it. We extract the DNA. We could PCR, do the PCR right there in the can, probe it, and take a picture of it and send it back remotely. Does it all in the can. You don't have to do anything, right? It does the sampling and the analysis and everything for you. We've now shrunk that whole thing because of 3D printing. If any of you are into 3D printing is great, because now you can start to miniaturize things, right? Microfluidics. You could build little tiny things with 3D printing. So we've taken that lab in a can and a 55-gallon barrel, and we'll put it into our long-range AUV, and the barrel is this diameter. And instead of that tall in the 55-gallon barrel, it's that long. same instrument, but different mechanics, different engineering, and a different processing, but the same idea. We could take the sample, extract the DNA or the RNA. We could amplify it, we could probe it, take a picture and send it back to us. And we also take an archival sample, just in case there's some anonymous reviewer who says, how do you know that was what it was? So we could do this, we could repeat it in the lab and do it the old fashioned way.
Awesome, thank you.
Yeah.
Well, thanks, Dr. Matsumoto.
Help yourself to a sticker and
Yeah, cool stickers up here, completed undescribed species, and he's also giving a talk in Wrigley Hall 481 at 4:30. Pizza.